Hello dear students, in this video we're going to talk about our new part of the nervous system. So first we studied about the parts 1, 2 and 3. These parts 1, 2 and 3 they are related to part 4, so you don't have to skip them with the notes, the parts you have to know it, the conclusions, all of them they are related to part 4. So let's start with 4. Part 4, in this part, we're going to talk about Francois Magendie experiment. This is the most important experiment through the nervous system. Let's talk about this experiment. So, experimental procedure. First of all, what did they do? They did the excitation of the ventral root of the spinal nerve. So, we have excitation of the ventral root of the spinal nerve. What happened? The concerned part, they contracted. So this means that the message moved from the ventral root toward the muscle. So the ventral root, it has a motor function since we have contraction of the concerned part. Excitation of the dorsal root in the second experiment. Excitation of the dorsal root of the spinal nerve. So what do we have? The concerned part contraction with a feeling of pain. So we have contraction and also we have feeling of pain. What does this indicate? This indicates that the nervous message was propagated from the dorsal root toward the spinal cord and from it toward the brain, and then it moves toward the concerned part. So the dorsal root, from here we can conclude that it has a sensory function. So here first we deduce that the ventral root, it has a motor function, since after the excitation of the ventral root and of the ventral root of the spinal nerve, we had the contraction of the concerned part, meaning that the nervous message propagated from the ventral root toward the muscle. So here the ventral root has a motor function, whereas in the second experiment, the excitation of the dorsal root of the spinal nerve, we had the contraction of the concerned part with the feeling of pain, meaning that the message moved from where it was propagated from the dorsal root toward the spinal cord and then to the brain and then toward the concerned part, which is the muscle. Therefore, it has a sensory. So dorsal, S, sensory function, whereas ventral, motor function. Let's move to part 3 of this experiment. Part 3, it talks about the excitation of the central end after the section of the ventral root. We did the section of the ventral root. So what happened? There is no reaction, meaning that there is a problem in the propagation of the nervous message, meaning that the message was stopped here. So stopped at this section, while after the excitation of the peripheral end of the ventral root, we have contraction. So here the message was propagated toward the muscle. Okay, so this means that we have a propagation of the nervous message toward the muscle. So it propagates in a centrifugal direction in the motor ventral root. While let's move for the sectioning of the dorsal root. So we did the sectioning of the dorsal root and we did excitation of the central end. What happened? We have contraction with a feeling of pain. So this indicates that the message propagated toward the spinal cord, then to the brain, then to the muscle. While after sectioning the peripheral end, there is no reaction, meaning that the message was stopped at this section. So here the message stopped. So here we can conclude that the message here propagates in a centripetal direction, meaning that it moves toward the spinal cord and the brain, where in the sensory dorsal root. Okay? While the last experiment, after we destroyed the spinal nerve, we had total destruction of the spinal nerve. So we destroyed the spinal nerve, there is no reaction, and we have total loss of the motor activity. What does this mean? This means that the spinal nerve, it has mixed function. It has both the sensory and the motor. It has both sensory function and the motor function. So let's repeat this. After sectioning of the ventral root, what happened? After sectioning of the ventral root and the excitation of the central end, there is no reaction. So the message stopped here. While after excitation of the peripheral end, we have contraction and we have normal sensations, meaning that it moves toward in the ventral root, meaning that it moves toward the muscle. So it moves in centrifugal direction in the motor ventral root. What did you say motor? Because it has a motor function. While in the dorsally, why do we call it sensory? Since it has a dorsal function as explained in the second experiment. So sectioning of the dorsal root. First, we have excitation of the central end. So we have contraction with feeling of pain. So here we can conclude that the message propagated to the spinal cord, to the brain, and then to the muscle. While after sectioning of the dorsal root and excitation of the peripheral end, what happened here? There is no reaction. So we can conclude that the message propagated in a centripetal direction toward the center, toward the brain and the spinal cord. So here we can conclude that the nervous message in the sensory dorsal root propagates in a centripetal. While the last experiment, which is experiment number 5, total destruction of the spinal nerve, there is no reaction, there is no sensory, no motor, total loss of motor activity and sensation. So there is no sensations, there is no motor activity, so we can conclude that the spinal nerve, it has a mixed function, both motor and sensory. Why? Because when you destroy the spinal nerve, there is no motor or sensory function. Okay, this is the most important experiment in the nervous system, in the neurophysiology, which is the experiment for Magendie. Okay, so they said interpret the results, deduce the role of the dorsal and the ventral. So interpretation with deduction. So after excitation of the ventral root of the spinal cord, the concerned part, it contracts. This indicates that the nervous message was propagated from the ventral root toward the concerned part, which is the muscle. While, therefore, the ventral root, it has a motor. While after the excitation of the dorsal root of the spinal cord, the concerned part contracts and the animal feels pain. What does this indicate? This indicates that the nervous message was propagated from the dorsal root to the spinal cord, and from it to the brain, and then to the concerned part with the muscle. Therefore, the dorsal root, it has a sensory motor function, while the ventral root has a motor function. Number 3, after sectioning the ventral root and exciting the central end, there is no reaction. So the nervous message stopped at this section. While after sectioning the ventral root and exciting the peripheral end, the muscle contracted. So the nervous message propagated toward the muscle. What does this indicate? This indicates that the nervous message propagates in a centrifugal manner, and the motor ventral root, it moves toward the muscle. These conclusions, you can study them in order to know, in order to realize the different properties of this experiment. You can study them. You can study that, for example, the sensory nerve after excitation of the sensory dorsal root, you can study them. You can study that the dorsal root or the ventral root, for example, first one, the ventral root has a motor function. You can study that the dorsal root has a sensory function. You can study that the nervous message propagate in a centrifugal manner in the motor ventral root, and after sectioning the dorsal and exciting the central end, contraction occurred and the feeling of pain was recorded. What does this indicate? This indicates that the nervous message propagated to the spinal cord, to the brain, and then to the muscle. Yet, after sectioning the dorsal root and exciting the peripheral end, there is no reaction. This indicates that the message stopped at the section. Therefore, this one you can study it. Therefore, the nervous mass propagates in a centripetal manner toward the center, toward the brain, to the spinal cord, and the sensory dorsal root. Last experiment was destruction of the spinal nerve. So we destroyed the spinal nerve. So there is no reaction and there is total loss of motor activity and sensation. What does this indicate? This indicates that the spinal nerve has a mixed function. It has both sensory and motor functions. So spinal nerve has both sensory and motor functions. Here move for the landline phone. What does the landline phone works with the biology? Landline phone. This is the first time you hear about this. Do we have phones? Do we have landline phone in our body? I didn't hear about this before. So landline phone. We have neurons inside our body, right? These neurons, they are these two men. They are talking to each other using the landline phone. The connection between them, it's called synapse. So the connection between two neurons, between two people talking to each other, sending messages between each other, calling each other, it's called synapse. So what is the synapse? How does a nervous message propagate from one neuron to the other cell? Let's talk first about the synapse. Synapse is like a link between a neuron and another neuron, maybe it's another muscle, maybe it's another gland. So synapse, it's like a link, it's like a connection. So what definition of a synapse? Is the functional connection between a neuron and the other cell. It doesn't mean the other cell, it's always a neuron. Maybe it's a neuron, maybe it's a gland, maybe it's a muscle. So, synapses. First, presynaptic neuron. Pre, it's before the synapse. So this one is called the presynaptic neuron, while the other is called postsynaptic. So before, it's called the pre. Before the synapse is called pre, and after the synapse is called post. Okay? So presynaptic, postsynaptic. So before the synapse, it's called pre, post, it's after, and the middle, we have the synapse, and we have something called synaptic cleft. It's the region where some chemical messengers, we're going to talk about them later, they are secreted or they are released into this cleft to cause a reaction. The synaptic density. Synapses, they are created with astonishing speed in the first 3 years of life. For the rest of the first decade, children brains have twice as many synapses as adults, and this is shown here between 6 and 14. Classification of synapses. Synapses are classified either according to their structure or according to their function. According to their structure, they are classified as neuronal synapse. What's the meaning of this? Its name indicated that neuronal is between a neuron and another neuron. Neuromuscular synapse is between a neuron and a muscle. Neuroglandular synapse between a neuron and a gland, for example, adrenal gland. Okay, so these two, they are called neuroeffector, between a neuron and a effector organ or muscle. Okay, so according to their structure, here we have neuronal synapses. So these are between two neurons, or neuroeffector synapse, either between a neuron and a muscle or between a neuron and a gland. So neuronal, as I said, between two neurons, and neuroeffector between a neuron and a muscle or between a neuron and a gland, meaning that between a neuron and an effector. Neuron synapses, these are example about them. Neuron synapses. Some of them, neuron synapses, are classified also into different forms. Either they are called axodendritic. Axodendritic between the axon of the first neuron and the dendrites of the second neuron. Or it's called the axosomatic between the axon of the first, this is the axon of the first, and the soma of the second. So it's called axosomatic synapse. So this is the second type. The first one was axodendritic between the axon and the dendrites. The second one is called axosomatic between the axon and the soma, as shown here. This is the axon, and here we have the soma, or it's called the cell body. Third type is called axonal. It's between the axon of the first and the axon of the second. So axon of the first, axon of the second is called axonal. Neuroeffector synapses, they are classified either neuromuscular synapse, motor, and a plate. So neuromuscular synapse is called the motor and plate between a neuron and a, or neuroglandular synapse, it's between a neuron and a gland. So as I said, neuroeffector, they are divided into two types. Either neuromuscular synapse between a neuron and a muscle is called motor and a plate, or neuroglandular between a neuron and a gland. According to their function, either the synapses is going to be inhibitory or they are excitatory. Inhibitory, they inhibit the message. Excitatory, they allow or permit the message to propagate. So first, definition of excitatory synapse. Excitatory synapse, they allow the propagation of a nervous message from the presynaptic neuron to the postsynaptic cell. Why did I say neuron? Maybe it's a neuron, maybe a muscle, or it's maybe a gland. So excitatory synapses, they allow or they permit the propagation of the nervous message from a presynaptic to the postsynaptic. Inhibitory synapses, what do? They do the opposite of the excitatory, so they inhibit the propagation of the nervous message, meaning that they stop it, or they block it, they prevent the propagation of the nervous message from the presynaptic neuron to the postsynaptic cell. Transmission across the synapse. Transmission across the synapse occurs from the pre to the post. In the post, we have the postsynaptic membrane, where some messengers they will bind. So here, detail of the axon terminal. This axon terminal contains synaptic vesicles. Synaptic vesicles, they contain special chemical messengers. These chemical messengers, they are called neurotransmitters. They are released from the pre to the post in the synaptic cleft, where they bind to the postsynaptic membrane through the binding sites. Here we have the presynaptic membrane. We have the synaptic vesicles that contain the neurotransmitter molecules, like for example acetylcholine or dopamine and others. They are released here in the synaptic cleft and they bind to the binding site on the postsynaptic membrane. How binding sites work? Let's see. So here the neurotransmitter is released, it binds to the binding site and causes the entrance of ions like Na+ ions. Okay, as shown in this figure. Propagation of a nervous message through a synapse. Let's talk about the neurotransmitter. What is the neurotransmitter? The neurotransmitter is the chemical mediator between two synaptic cells. It's like a connection. It's like a, for example, a taxi driver between to synaptic cells. It's like a chemical mediator. It helps like a messenger. It's released by the presynaptic neuron. It's released by presynaptic neuron, and also it's released in the synaptic cleft where it binds specific receptors where on the membrane of the postsynaptic cells. So whenever it binds on the membrane of the postsynaptic cells, this will cause the reaction to occur. Propagation of a nervous message through a synapse. First, the nervous impulse reaches the presynaptic terminal, causing depolarization. Then, this depolarization will open the voltage-gated calcium channel Ca2+, so it will rush into the presynaptic. Then, the secretory vesicles will be mobilized toward the cell membrane of the buds and they bind to it. What do the secretory vesicles contain? Then the vesicles will release the neurotransmitters. So these vesicles, these sacs, they have neurotransmitters into the synaptic cleft, so they release them by the process of exocytosis. So this neurotransmitter will bind to the receptors on the postsynaptic. If the synapse is excitatory, the neurotransmitter will bind to the chemically gated Na+ channels. So the Na+ channels will open and Na+ will flow into the postsynaptic cell, causing the depolarization. It's called EPSP, excitatory postsynaptic potential. EPSP. So as I said first, the nerve impulse will reach the synaptic terminal buds, causing depolarization. This depolarization opens the voltage-gated Ca2+ channels, so they rush into the presynaptic buds. Number 3, these vesicles will move toward the cell membrane of the buds and will bind to it. These vesicles will release a neurotransmitter into the synaptic cleft by the process which is called exocytosis, and they will bind to their specific receptors on the postsynaptic cell. There is 2 cases: if the synapse is excitatory or if the synapse is inhibitory. If the synapse is excitatory, what will happen? If it's excitatory, the neurotransmitter will bind to the chemically gated Na+ channels. So Na+ channels, they will open, Na+ will flow into the postsynaptic, causing depolarization. So it's called EPSP, excitatory postsynaptic potential. While if the synapse is excitatory, this depolarization exceeds the threshold. If this depolarization exceeds the threshold, then an action potential will be violated. While if the synapse is inhibitory, the neurotransmitter will bind to the chemically gated K+ channels or to the Cl- channels, chloride channels, or potassium channels. As such, these channels, they will open. So the K+ will flow out of the postsynaptic cell, or Cl- will flow into the postsynaptic cell, causing hyperpolarization, which is known by IPSP, inhibitory postsynaptic potential, IPSP. So just due to this hyperpolarization, no response will be recorded in the postsynaptic. In both synapses, in order not to expose the postsynaptic cell, the neurotransmitter will be degraded by enzymes. So these neurotransmitters will be degraded by enzymes to not expose the postsynaptic cell by their action, and this will help the postsynaptic cell, or this neurotransmitter will be recaptured by the presynaptic cell.